different DNA
Okay, let's have a class, and I will be real quick to go over the left slides. The majority of the time we're going to do the review, go over some review questions. Next week, Tuesday, we do not have a lab, then we will have a lecture exam 2 right here. And the key I will put on the eCampus over the weekend. Now, by the way, by the noon today, I have to put on the midterm grade, but it does not really matter. It's just a reference. Okay, so don't worry about that too much. Even you get a D or C, you still have a chance to get a B or A in the final exam. Don't worry about that too much. Okay, radiation. Everybody knows that ultraviolet light can be used to control bacteria because they will form some dimers. That will prevent the DNA replication. And the X-rays, gamma rays force electron out of the microbial molecules. Okay, so you can see from here, even the sunlight, there is a little bit area which is sunlight right here, which is a little bit of area here, which is still be sensitive to killing bacteria. And the X-ray, gamma rays, these ionization short wavelengths typically will take out of the electron from the microbacteria, which means prevent the metabolic process, lose the energy. There is some of the products could use food irradiation as a preservative methods. And a very good example will be the ground beef. Ground beef requires zero tolerance for E. coli 057H7. So some of the plants, when they come out of the plants, the last step they do is screening with X-ray radiation, and they have to be approved by the National Aeronautics and Space Administration, NASA, and also you have to be certified by that. Okay, not everything you can do, just do a radiation. But keep in mind, the radiation message is only on the surface. It's not really penetrate too much. This slide tells you is if it's an ideal disinfectants, what they will be achieved for what the requirements of these. They should be killing bacteria. They are non-toxic to the human, to animals. They can be easily dissolved in the water, and it have a longer shelf life and will be very effective in a short contact time and with low concentration. And we are very important to choosing the agent, which is the temperature, pH, and how long it's going to do the disinfection. However, I would love to tell you there is no such chemicals in the world so ideal and nice to everything. This is talk about is cleaning and sanitizing. So cleaning, we really talk about removing food soil, which is fat, protein, carbohydrates, and mineral salts. So first of all, when we talk about the cleaning, we talk about detergents. Detergents is not really antimicrobials. Lots of the time, your detergents is treated as antimicrobials because there is antimicrobial ingredients there. So detergents, the first component is surfactants. Surfactants will be extended the contact area of the detergent itself. Also could be breakdown. The large molecules such as fat become small fatty acids. That's called emulsification. And usually it's built up by alkaline acid, enzyme, or some oxidizing agents. That's called a builder. And then we would very much want it to be easily to handle. Therefore, we have a filler. The filler usually it's a water or salts. Typically, the salts is very nice because it can be dissolved in the water. And we don't want them to be alcoholic material because if it's a alcoholic material, especially like oil type items, they cannot dissolve in the water too much. Another thing, the last thing, usually of those components, they are including the corrosive inhibitors because we don't want a corrosive happens. So basically what they do is a physical interaction with food soil, chemical interaction with food soil to form the water-soluble substances. This is the cleaning we want to mention. There is two products right here. Is Zephyrin and cetipyridine chloride. This second one, which is we call it a CPC. This is a product widely used in the poultry industry, which is before and after chilling. It's a very strong antimicrobial impact, but it's like a bubble. Okay, it's like a soap bubble, sometimes looks like. They cause a leakage of cytoplasm, and they will be reacted with cell membrane, destroy bacteria and the virus. Now, very important you need to know, all these antimicrobials or chemicals, they have to be regulated by 3 agencies. Number 1 is EPA, United States Environmental Protection Agency. They will do the evaluation to see the products is environmental friendly. So they will look at your antimicrobial efficacy data, chemical components, is there toxication for the environment, and how you label it. Now, once the food products can be used by these antimicrobials, all these sanitizers, they have to be certified or approved by 2 agencies, FDA, US Food and Drug Administration, and the USDA FSIS, which is Food Safety and Inspection Service. USDA, which is United States Department of Agriculture. They need to be allowed, these EPA approved sanitizer could be used on the food contact surfaces. Here are the examples. In 1996, approved by FDA, all these chemicals. So we want to go these 2 details. Some of these I do a lot of research. First of all is chlorine-based sanitizer, which is chlorine. Chlorine, basically, we talk about is hypochlorite acid. So, said very simple. Working with water, then become what? Is this guy. We really want is this guy. This is called free chlorine. However, this free chlorine is very sensitive to organic matter, which means debris, dusts in the container. If there's an organic matter there, it will be lowered up 50 parts per million free chlorine concentration to less than 0.5 ppm in roughly about 30 seconds. And the research has been showing you have to be maintain the free chlorine concentration at least 10 ppm to prevent cross-contamination in the fresh produce. And very important that free chlorine concentration you have to control. And also you need to make sure this pH has to be roughly 6.8. So industry, they use citric acid to buffer the pH. The pure chlorine pH is usually around 9 to 12. It's an alkaline status. We need to buffer to the neutral pH, 6.8 use citric acid, then the free chlorine will be the dominant components. Now the free chlorine will be reacted with organic matter. Then the concentration will be decreased. So what the industry they do? They refresh, re-added. However, if we re-added, what's going to happen? We will be generating a lot of chlorine off gas if the pH is going to be lower 4. And then they will generate lots of the chlorine byproducts. The byproducts like trihalomethane, PHM, trihalomethane, all these kind of chemicals. And I have the research which is tested during the produce washing, how many of these byproducts going to generate. Because of this reason, so the free chlorine concentration is not allowed to be using during the fresh produce washing in European country. They can only using they recommended to use is electrolyzed water. We will mention later on. And sometimes they require to use PAA, peroxyacetic acids. So just very briefly here. Chlorine, because chlorine is easy to react with organic matter, so people comes out with chlorine dioxide. But the problem is chlorine dioxide is a gas, so it's very difficult to control. Sometimes we need to do something which can be monitored. So there is a color. So what we use? Iodine. We use iodophones. Iodophones is a brownish color. We know we added it. The major common sanitizer used in the lab and in the processing plant is contrary ammonium compounds. We call it COTS. They also have some acid-based products. That's carboxyacidic sanitizer, acid anoic sanitizers. But be careful. Sanitizer with acid-based is very corrosive. And lots of the metal could be caused decay. Peroxyacetic acids hydrogen peroxide they can combine, which is a chemical called sanidate. I'm doing a lot for research for that. In the West Virginia local communities, they do a lot because they are kind of organic certified. You can use it in an organic process. I'm not going to go all these one by one, very detailed, but there's a keep in mind. When you're choosing a sanitizer on the food contact surfaces, the key is you have to pick 2 sanitizers and do a rotation. If you stick to use one, it's very important the bacteria will become resistant. And once the bacteria become resistant to one chemicals, it will generate cross protection. So let's say if you use acid-based sanitizer, the bacteria become acid resistant. Then it needs much more concentration of PAA if your second treatment is PAA. Therefore, you need to keep doing the rotation of those 2 sanitizers to avoid or diminish of this type of the resistant bacteria comes out. Okay, so that's a key. Next, we want to be very simple to go over these. Phenolicus, this is the first and the oldest antimicrobials in the world. People use it. They're using a hospital disinfection laboratory and basically denaturing protein and disrupt the cell membrane. However, it will cause a very strong skin irritation and a disagreeable odor. That's phenolics. Alcohol. Everybody keep in mind, 100% alcohol doesn't work for curing bacteria. You need to dilute it in the water, at least 95%. The ideal is 70%. Keep in mind, 100% alcohol never kills bacteria. You need to dilute to 70 to 95%. It will be killing majority of the bacteria. Inactivated virus may not be very workful for the endospore, but it's very effective. We call it disinfectants. So the nature proteins possibly dissolve membrane lipids. This is CPC, which is cetapyridine and beta-carotene. It's widely used in the poultry industry we just mentioned. It's an organic cleansing agent. Sometimes it's used for killing bacteria. However, Microbacterial tuberculosis and endospore bacteria, which means Clostridium bacillus, it doesn't work very well. This is easy to use, easy to prepare. However, make sure you do not use hot water. What is hot water? Calcium and magnesium reach certain level, 100 ppm. You have to make sure the hot water, you have to become a soft water. So how to do it? The easiest method is boiling it. If it's temporary, calcium and magnesium there. So give you examples. A pound water, a well water is hot water. And your drinking water, municipal drinking water is usually soft water. Okay, so just be aware of that. Aldehyde. Formaldehyde, or glutaric aldehyde, those things usually don't use on food contact surfaces. They are using to preservation in the museum, which is all those animal samples, all those plant samples. So not used too much in our everyday life. Therefore, inactivating nucleic acids and the proteins. By all saying this, they also have other biological control of bacteria. We could generate lactic acid bacteria. Lactic acid bacteria will generate lactyricin. It will be killing bacteria. Okay? We could use bacteriophage. Bacteriophage is specific for certain bacteria, which is a virus to eat a bacteria. Some of them, the genome will be integrated. We call it a warm phage. Some of them are temperate phage. Some of them will cause bacteria died. We call it a lytic phage. So phage will be one of the way we can use. And some of the chemicals. Okay, so, and the bacteria. So just want to let you know about this. This very briefly, we talk about this. Okay, we're going to talk about the exam questions and do some of the review. Very important, the major part of exam 2 will be the bacteria pathogen caused disease. So you have to study very well. What will be the question looks like? I will be open this one first. in the exam, we do not require you to write a bacteria name. The question will be very similar to right here. At the beginning of the exam paper, I will give you the name of all these bacteria. Corresponding to a letter, you only need to pick from there which bacteria it is. The key thing is the case study. Some of the case study will be very similar to here. Some will be new. Some of them will come in from the after class reading. So be careful to read all those. Okay, let's talk about all these questions. The first question. John is an English instructor at the university. Received a call from the adult daycare. Said 74-year-old mother was staying. Experienced chest pain, chill, had a fever. Arrived at daycare, she seemed to be short of breath and cough, and had mornings that worsened and become productive cough. The local hospital prepared a sputum sample grown blood agar and chocolate agar have alpha hemolytic zone. And the gram stain results, it's gram-positive diprococcal, catalase test negative, and the optically susceptible test. What is the bacteria it is? Which bacteria is gram-positive diprococcal from these? And the catalase test negative. So make sure, this is you understanding. Staphylococcus aureus versus Streptococcus. The first thing first is catalase test. Negative and positive. So it's a Streptococcus. And the gram-positive diprococcal, which is alpha hemolytic. When we talk about alpha hemolytic, what it is? Viridian Streptococcus could be. Another one, Streptococcus pneumoniae. Is that right? And this is typically fit for gram-positive diprococcal. So therefore, the answer here, Streptococcus pneumoniae. So easy. A. Okay, that's second one. Look at the information here. That's 4-year-old kid. Is that right? What you find? Headache, photophobia. Then we find this bacteria. Find it only grow on chocolate agar. What this means? It's a fastidious bacteria and need XV factor to grow. And the symptom here, very typical, looks like is a meningitis. Which bacteria cause meningitis in 4-year-old kids? Can you tell me? And need X and the V factor? Haemophilus influenza. And remember, 0 to 2 months, Streptococcus ag lactea. 2 months to 5 years old, Haemophilus influenza. And typically type B. Okay, how about 5-year to 40-year-old? Neisseria meningitis. Okay, 40-year-old above, Streptococcus pneumoniae. So when you study this question, these things you need to know. Okay, it's so simple when you do it. Okay, 27-year-old female. He not feeling very well, have a fever, developed a rash, and experienced a sudden dropping of blood pressure, kind of like a shock symptoms. The blood of the cerebral spinal fluid was negative for sepsis and the bacterial meningitis. His history was significant. He is experimentally using a new contractive sponge that has not been removed since the previous evening. Which one will cause this problem? This is same as a highly absorbent tampon. So what is one called this? Called this one. This is a typical symptom for TSSI, toxic shock syndrome toxin, caused by Streptococcus aureus. Okay, so you're gonna have to pick the Streptococcus aureus. A gram-positive coccyl was isolated from 37-year-old female UTI, urinary tract infection. And the lab test found this grown blood agar, no hemolytic zone, catalase test negative, belonging to Rebecca Lansfield group D. Okay, we're going to Rebecca Lansfield group. What are they? Group A, Streptococcus pyogenes. Group B, Streptococcus agalactia. Group D, what is that? Enterococcus facilis. Okay? So you're going to pick the one which is Enterococcus facilis. A 22-year-old sexual active female go to student clinical center, complain painful and frequently urination, that's called dysuria. And technician do the urine sample, find a gram-positive coccyl grape-like cluster. It is Staphylococcus, it tells you. They are resistant to novel vaccine. Here is going to be the key. Staphylococcus aureus, Staphylococcus epidermis, and the Staphylococcus agalactia. Saprophyticus. Which one resistant to the novel vaccine? Is that right? Low concentration novel vaccine. And Niconium honeymoon bacteria. Okay, Staphylococcus saprophyticus. 29-year-old pregnant females, they go to have a home birth delivery with a midwife. Not happen too often these days, okay, very few. Delivered a healthy 7-pound baby boy. On the day 5 postnatal, he refused to breastfeeding and have a fever 105. Okay, it's a gram-positive, catalase test negative, beta hemolytic. And this bacteria resistant to the base tracing. What is the components of the base tracing? A disca. What are we talking about the A disca? Agalactia. And what the other one? Pyogenes. This guy resistant. And the pyogenes susceptible. Always say sensitive, okay, so susceptible. So, which bacteria it is? Especially this is a newborn baby, kind of like a fever, vomiting. This is group B Streptococcus agalactia. Okay, next one. 5-year-old crossed into United States, a freight train from Mexico, but were apprehended by U.S. immigration authorities. These kind of sentences, I maybe have to took them out these days. It's too political, is that right? That's a question which is published like 2010 something. But it is a true story. And you find that difficult swallowing, neck swelling, blackish green, membranous, and then you see it's a gram-positive, club-shaped arrangement. And will be cultured on the blood agar with telluride. It's telluride blood agar. So what it is? What is a ball neck? The greenish, blackish greenish membranes, pseudomembrane system, and then you have a club-shaped gram-positive arrangement. What is that? Corynebacteria diphtheria. Is that right? Corynebacteria diphtheria is gram-positive club-shape. Basically, for immigrant kids go to United States because they do not have a vaccine. They didn't take Dtap vaccine, diphtheria, tetanus, and pertussis. Okay. Number 8 is easy. From your pathogen list, which organism is the most post-surgery infection? And belongs to a group of antibiotic-resistant bacteria named as MRSA. This is easy. MRSA is methicillin-resistant Staphylococcus aureus. So that's the Staphylococcus aureus. Okay, this question. Yavapai County, Arizona, a 13-year-old boy in 8th grade was cultured confirmed positive for pertussis and had attended school during his illness. To identify others why they may be infected with a probable clinical confirmation was established for the other students in that middle school. What's the criteria for the clinical confirmation? So what is the disease of this called pertussis? Whooping cough, is that right? So what do we do the testing for the whooping cough? PCR methods and nasopharyngeal specimen. Remember I said the nasopharyngeal specimen going to a nose should be half and between the distance to the earlobe and the nose. So the answer is B. Okay. The culture-based screening method for prevent systematic GBS infection recommends to what? Screening pregnant women for vaginal or rectal GBS between 35 and 37 weeks of gestation. So the answer is C right here. The approximate distance you should insert a nasopharyngeal swab is half the distance between nose and earlobe. Okay, from the pathogen list, which organisms are now considered part of the United States recommended child immune schedule? What are they? HIV, let's look at that. What is A? Streptococcus pneumoniae, yes. Second one. So A is covered. How about L? Let's look at the L. Streptococcus saprophyticus, no, there's no vaccine for that. So A is gone. How about M? Where's M? Haemophilus influenza, yes. HIV virus, we have it. So that's B. How about O? Corynebacteria diphtheria, yes, we have it, DTAP, and the Bordetella pertussis. So the answer here would be AMOP, which is C. Okay, last one. When etiological agent is responsible for child paragenesis, tonsillitis, sinusitis, otitis, and suppurative skin infection with sequelae such as acute rheumatic fever and glomerulonephritis, what is that? That is a bacteria called strep throat, because there is a very severe sequelae is rheumatic fever and glomerulonephritis, which is a transmembrane system which is damaged in the kidney. So what is that? Streptococcus pyogenes. So the answer is D. Okay? So these questions you have to practice. And then go back to look at the class videos or notes, what they are related to. Okay, question number 2, this set of the question, is a very simple test, which is the same thing, pick from the top. And then you're going to choose based on these description. It's very simple for you to review all these questions. So let's go over these. Which bacteria can grow on bioasculin agar, hydrolyze bioasculin, become bioasculitic? Group D Streptococcus. Remember? Become asculating. And it's a black colony. And it's in a bioasculating agar. And it's also resistant to 6.5% salt. Okay, so that's the first one. Second one we just mentioned. Can produce toxic shock syndrome by elaborating a toxin referred to TSST1. What is that? Staphylococcus aureus. That's simple. J. Pathogen forms of may produce beta-hemolysin enzyme coagulase. Same thing, these three. Which one have a coagulase test positive? Coagulase, which means you cause blood clotting. So which one? Staphylococcus aureus. Okay, will cause blood clotting. Catalase tested negative, gram-positive cocci. When you see this, it's Streptococcus. Belonging to immunological group A. Okay, that's easy. Streptococcus pyogenes. It's group A streptococcus. And susceptible for bacitracin. Bacitracin, which is an A disc. Okay, next one. Alpha hemolytic gram-positive cocktail. Catalyst test negative, sensitive to bile salts and other surface areas such as optican. Then what is that? Which one is sensitive to optican? Catalyst test negative, gram-positive cocktail. It's streptococcus. And then they are sensitive to optican. Then it's streptococcus pneumoniae. So the following. Number 6, a gram-positive cocktail that is catalyst test positive, coagulase test negative, and resistant to low concentration of novel biocin. We just mentioned honeymoon bacteria, Streptococcus, Staphylococcus saprophyticus. So will be K. Okay, saprophyticus. Can grow on and ferment lactose or McConkey agar. There will be more answer, which will be more than one answer. E. coli is one of that. What else? Calabacella ammonia. Remember, we have a picture there, Calabacella ammonia. We see there is a very cheese-like colony because mucoid colony, and very heavy capsule because, and it's also grow on the McConkey agar fermented lactose. That's why the colony is pink. Number 8, this microorganism can grow on selective media containing 7.5% sodium chloride, does not coagulate rapid plasma, and is susceptible to novel biocin. Okay, that's going to be, cannot do the coagulate. Can grow on selective media containing 7.5% salt. What is that? Does not coagulate, susceptible to novel biocin. Very possibly this one is Staphylococcus epidermis. Is that right? Because not coagulated rapid plasma susceptible to novel biocin. So Staphylococcus epidermis. Number 9, belonging to a group of alpha hemolytic microorganisms that normally live in the upper respiratory tract and may cause opportunistic infection. Alpha hemolytic microorganism. Here, cause osmotic infection, usually coming from oral cavity. That's Streptococcus, Viridime Streptococcus. The answer is C. Okay. Is a gram-positive endospore lot found in the soil. What is that? Bacillus? It should be the Bacillus subtilis. This gram-negative lot grows rapidly on blood and chocolate agar, producing a mucoid colony and the fermented lactose on McConkey agar. We just mentioned the mucoid colony, which means a heavy capsule, and the fermented lactose on McConkey agar, which means it's a pink colony on the McConkey agar. So it is Calabacella ammonia. If you forgot about this, you should go back to look at the slides. It will be a slides right. Calabacella ammonia. If you forgot about it, we're gonna go back all the way here to look at the slides. Is this guy, Calabacella ammonia. Blood agar on the side, McConkey agar on the left. What you see? Well, you're on the opposite direction. That is on the left. Left is McConkey agar. Pink colony, mucoid colony, lactose fermentation positive, Calabacella ammonia. So that's the answer where it is. Okay. So we finished talking about here. You don't have to worry about that one. This one I didn't mention. Fastidious negative lots that only grow in BG agar. It takes 7 days to colony forming. This Haemophilus influenza. You don't have to worry about that. The pathogen cause whooping cough, Bordetella pertussis. You know about that. Okay. Oh. Gram-negative lots that grow on chocolate agar, not on blood agar, which means it's a fastidious bacteria. Usually causing meningitis and epiglottis in age 2 to 5 years old. What is that one? Haemophilus influenza. It's 2 months, 5 years old. Kids have meningitis. Okay, responsible for the toxigenic disease can produce a thick greenish pseudomembrane in the back of the throat and a lymph and endosophagy known as bull neck. Which one has the bone neck and the thick greenish pseudo membrane in the back of the throat? We just mentioned Corynebacterial diphtheria. Okay, so you just find the answer here. And keep in mind, these choices could be multiple used. You're not going to be using only one time. Okay, Corynebacterial diphtheria. HIV vaccine is for this pathogen. What is that? Haemophilus influenza. So I would suggest you, when you do the review, don't put the ABCD there. Put the bacterial name there. Then you know which one is which. Okay, don't just put the ABCD, put the bacterial name there. The Dtap vaccine contains a toxoid to prevent which pathogen from cause disease. That's a multiple choice. What is that? What is D? Corynebacterial diphtheria. What is T? Clostridium tetanus. A is acellular and P is pertussis. Be careful, you know about this. Corynebacteria, diphtheria, Clostridium tetanus. A is acellular. You don't have any meaning, okay? Bloodterra pertussis. Just be aware of these. You don't have to write. We already tell you. You just pick the ground choice. A gram-positive catalase negative coxae that belongs to lungs field group D. That's easy. Enterococcus facilis, right there. Can infect the skin and cause impetigo or deep cellulitis. Remember, at the beginning, impetigo is on the surface. Then the cellulitis a little bit deeper. Sequela following actual pharyngeal infection, including rheumatic fever and glomerulonephritis. We just mentioned, what is that? Strep throat, Streptococcus pyogenes. Okay, here is another thing what you do. When you do these questions, write your name on the left side. Then you're going to can review. Also, you get another piece of paper. How many of these are repeated happen? And then you do another conclusion. So for example, there is maybe 3 questions. Is same for Streptococcus pyogenes. There is maybe 2 questions or same for Corynebacterial diphtheria. You got to conclude them. And our test exam questions will be highly likely looks like that. It's just a couple of words sentence. It doesn't matter. But the key information will be there. So this piece of sheet is important for you to do the conclusion there. And that's why I want you to practice these questions. Okay? So that's the second one. The last one, we want to also go over it. Okay, some questions right here. We want to talk about these. How you differentiate food infection and food intoxication? That's so simple, is that right? Food infection, you're eating bacteria. Food intoxication, you're eating toxin. What is a food infection? E. coli 157H7, what is food intoxication? Bacillus, Clostridium, even Staphylococcus aureus. How we generate the decay formation of the teeth? Do you still remember in our teeth area what happened? There are some of the sticky of the sugar there, okay, in the fermented environments. Streptococcus mutans. So let me draw it for you. So teeth, okay, sticky residue sugar. Okay, then you have a Streptococcus mutans. When the Streptococcus mutans are in the anaerobic environments, we're going to go through fermentation. Okay, what's the fermentation? What's going to happen? The acid will generate, is that right? Acid comes out. NML. So de-anemorization will cause the teeth decay because acid will have some impacts for the teeth. Okay, that's not difficult. Okay, what is the difference between botulism and the tetanus toxin? Name 3 types of botulism. What are 3 types of botulism? Foods, woods, and infant. What is the difference between botulism and tetanus? Botulism, what you have is a floppy baby, is that right? Tetanus, what you have? Lockjaw. So one is prevent contraction, another one is prevent relaxing. So one, the target is what? Chlorine, another is acetylchlorine. Okay, if you forget about this, go back to look at your slides. The slides will be somehow right here. Not this one. There will be somehow right here. This will be the answer. Look at that. Acetylchlorine and the glycine. There is a well-explained botulism toxin blocks release of the acetylchlorine and tetanus blocks release of glycine. Okay, so that's a very good picture there. Lyme disease, we have one here. Describe why vibrochloral causes patient dehydrated. What types of food will be possibly contaminated? What type of the food? There is, well, the seafood will be one of that. Jellyfish is one of that. What's a chloral will cause dehydrated? This will be found right here. So what it is? Sodium removed to maintain osmotic balance, and then the activate cyclase, so the ATP becomes AMP, and the highly cyclic AMP level block uptake of sodium, increase sodium chloride in lumen jaws in water. There lots of the water will come out. Okay, so that's a picture. You can do a brief description about that. Question number 5. Lyme disease is spread by mosquito vector. No. What is that? Ideldex, not mosquito. The disease caused by Streptococcus mutans is what? Dental decay or caries. Those who may be at a great risk for Shigella. Which one is for Shigella? We mentioned about for Shigella is the kids in the daycare because they go everywhere. They cause cover active Shigella toxin. So B will be the answer. Why are many diseases of gastrointestinal tract not treated with antibiotics? Because their stomach acid is so low, it will be decomposition, the components of the antibiotics. So answer is B. Okay. The primary treat for chloral. Since chloral, you will lose a lot of water, so what you going to do? Rehydration, is that right? The rehydration. Put back the water first. Okay, this is going to be the answer. Matching principles. Can you do that? Which one disrupts the membrane transport function? Is that F? Which one blocks transpeptidation reaction cell wall synthesis? Is that penicillin? Oh, sorry. It should be peptide transferase. That. Sorry, yeah, yes, it's A. Okay, so these two gone. You can mark it. Let's do. I may not remember everything. Let's do. Okay. Okay, now see. Inhibits DNA gyrase activity. What is DNA gyrase? That's one type of the topoisomerase. Okay, then we're going to put D. Natric acid. Okay, we have left those three, so we put this one. This revamping. Oh, sorry. There should be. Yeah, natric acid. Is that natric acid? How about DNA polymerase? Is revamping? Is that right? Okay, then revamping is gone. What are the two? Tetracycline and chlorophenicol. Okay, I don't remember it. Then we go back. So what we're gonna do? Go back to see number lecture 5. Okay, we'll go take a look together what it looks like. The last page will be right here. Which one? Tetracycline for tRNA. This is a little bit difficult, is that right? That's tetracycline E. Okay, which one you left? Chlorophynical is B. We'll see whether it is. Chlorophynical, okay. Yeah, prevent peptide from being bound. And then we're gonna be checking one more thing. Okay, what is that? Rifampicin RNA polymerase. So maybe I made a mistake here. Then we go back to fix it. RNA polymerase is rifampicin. Okay. DNA gyrase is D. Okay. Natric acid is one of the components of that. But I will be just change it to fluoroquinolones. Okay. This natric acid is so confusing. So that's fine. So these are the questions what we did. Which choices below represent 3 types of the botulism? Food, wood, and infant. Okay, that's easy. A. Now, go again. We have these questions. Again, I listed so many, then we're going to look at these. Okay, we're going to talk about these real quick. When you read all these, you see the swearing, double vision, slurred speech. This way you do. Highlight. Okay, that's kind of thing. Neurological symptoms. Then let's do canned food. Okay, which pathogen caused by canned food? Seems like Clostridium. Gram-positive large subterminal endospore. Okay, endospore forming bacteria from canned yellow tomato dish. What is possible? What is the pathogen cause canned food? Clostridium botulinum, is that right? So that's E here, so simple, because they'll give you so many information. 70 years, the question number 2, 70-year-old female developed a fever for a couple of days. So we just see this. No localization symptoms, large circular rash on the back. Okay. He is camping, biking on the Sorrel Hill Trail in Mammoth Cave National Park. That was in Kentucky. Around like 1 hour north